Okay, so I snuck off to do this video really quick. I'm a dietitian. My name is Meredith. I'm not your dietitian. I'm not your healthcare provider. So please talk to your healthcare provider before making any changes to your diet, your supplements, your medications, or your lifestyle. This video is specifically for anybody who has seems to be struggling with reactive oxygen species or oxidative stress, the connection really quick between folate and this pathway. So I'm going to share a diagram. Actually, I'm going to share the journal article. This is a journal article, and it's maintenance of cellular tetrahydrobiopterin homeostasis. Really nice journal article. I'm going to zoom in. I am going to show you this pathway. Actually, I'm going to use a different diagram in here. So this here is actually a diagram showing where a specific enzyme, dihydrofolate reductase, is involved in the recycling of BH4. BH4 is needed for proper function of nitric oxide synthase. Sorry for the background noise. It just came off. My kiddos are vacuuming and cleaning house for me. Okay, so here's DHFR. And so it can convert BH2 to BH4. And let me go down here. So you can see that that was to show you that it's an independent enzyme. It has the functional ability alone to convert BH2 to BH4. So here's the other diagram that I think is really interesting. This other enzyme, DHPR, dihydrooptidarian reductase, that's right here. It can also convert BH2 to BH4. Where is that up in the previous diagram? Let's scroll back up. That's this one, DHPR. So that's going from QBH2 to BH4. So you can see that DHPR is blocked by peroxynitrate, and DHPR is blocked by hydroxyl free radicals. Okay. Those can come from hydrogen peroxide, and that can actually come from taking superoxide dismutase and taking superoxide dismutase, the enzyme. Here's superoxide. Okay, so in vitamin A, hypervitaminosis A, there's a large amount of superoxide, and you can go back to a previous video to look at that. As long as you have functioning superoxide dismutase, then you're going to make hydrogen peroxide. Then from the Fenton reaction, if you don't have catalase on board, catalase is inhibited by excessive amounts of nitric oxide. So you'll end up with too much hydrogen peroxide. It'll go through the Fenton reaction and it'll make hydroxyl radicals, which will inhibit DHPR, and peroxynitrate will also inhibit DHPR. And so then what you would want to use is DHR, but that's also inhibited by hydroxyl radicals. So in this case, this is my crazy thoughts for the day. One thing I wouldn't do if I have excess superoxide because of hypervitaminosis A is I wouldn't take too much vitamin C, because vitamin C is directly degraded to H2O2 and can go through this Fenton reaction, and it's going to turn off your DHPR and your DHFR, and you need to recycle BH2 to BH4 so that you can keep INOS coupled. Okay. So that's one thought. I want INOS to be coupled. BH4 keeps INOS working well. So you can see that BH4 makes nitric oxide synthase become coupled, and coupled nitric oxide synthase does not make extra reactive oxygen species. So don't overdo vitamin C. There's a Goldilocks zone for vitamin C. The second thing about this is that if you're stuck in this whole pathway, you're not using folate properly. That's in a previous video, but basically your folate's going to be trapped. I will share this with you really quick. I wanted to make this so short, but you know me, I think this is still going to be one of my shorter videos. So if you have excess nitric oxide, then you're going to end up shutting off methionine synthase. This leads to a buildup of 5-methylfolate and a poor recycling of tetrahydrofolate. Most of us are probably having a functional B6 deficiency, so we're not going back and forth between 5,10-methylene tetrahydrofolate and tetrahydrofolate very easily. So we're going this roundabout way through 10-formal tetrahydrofolate, but most of us are having to use 10-formal tetrahydrofolate to take care of some of the glutamate issues, and we're making more uric acid in general. So if you have a gouty toe, that's what's going on there. So the problem is we're having to get more folate from our diet, and that's dependent on DHFR, which is already being impaired by excess reactive oxygen species. So do we want to put a bigger burden on DHFR to make THF? No, we don't. What we want to do is get a Goldilocks zone for folinic acid. I say Goldilocks because one of my kiddos has a folic acid deficiency, mostly because she's trapping 5-methylfolate. I gave her 800 micrograms of folinic acid, and now what's happening? Well, it's combining with her excess glutamate, it's becoming glutamine in the body, and she's getting gout in her hand that she likes to use the most. Her right thumb is having problems. Okay, so back to you. The other share, this excellent journal article, which I will put a link in the notes. So one, an oxidative stress, we're going to have decreased DHPR. It's more affected because more things can block it. Go back up. More things can block it. See, it's blocked by peroxynitrate and hydroxyl-free radicals, whereas DHFR is only blocked by hydroxyl-free radicals, OH, right? So what you really want to think about here is, do you want to tie up your DHFR in trying to incorporate more folate into your cells? One, you wouldn't want to do a folic acid supplement because that requires DHFR. Two, you probably want to do folinic acid. That would get you to where you need to be, where you need to add in tetrahydrofolate, but you don't want to go too high. Okay, so we need DHFR. Folinic acid bypasses that, so it won't put pressure on your DHFR enzyme. We need DHFR to recycle BH2 to BH4. Otherwise, we're going to have uncoupled iNOS or eNOS or any NOS, including mitochondrial NOS, because there's one of those too. So there's eNOS, iNOS, mitochondrial NOS, and there's one more. Oh, the one in the nerves. So the nervous system NOS. We want those all coupled. All right. So I think this was my fast video. And now I have to go help with the housework. Bye-bye.